In translation, amino acids are assembled into proteins. Bacterial translation is initiated in 3 steps. First, the small subunit of the ribosome binds to the mRNA so that the P site is positioned over the start codon. Second, the initiator tRNA with N-formylmethionine attached binds to the mRNA through base pairing between the codon and the anticodon. Third, the large subunit of the ribosome joins the complex to complete initiation. During elongation, amino acids are joined to a growing polypeptide chain. The first step of elongation is the delivery of tRNA and its amino acid to the A site. In the second step of elongation, a peptide bond forms between the amino acids. The third step of elongation is translocation, the movement of the ribosome down the mRNA. After translocation, the tRNA formerly in the P site is now in the E site, from which it moves to the cytoplasm. Now the elongation cycle repeats itself. A tRNA and its amino acid occupy the A site. A peptide bond forms, and the ribosome translocates to the next codon. As the cycles of elongation continue, the polypeptide chain grows by the addition of amino acids to its carboxyl end. The last stage of translation, called termination, takes place when the ribosome reaches a termination codon. Release factors bind to the termination codon, causing the release of the polypeptide chain. The ribosome then translocates one last time. The tRNA and release factors exit the ribosome, and then the ribosome dissociates. Remember that several accessory proteins not shown here also participate in translation. Let's design mutations in the coding region of a gene and discover their effects on the protein sequence. In our first example, let's change a C in the mRNA sequence to a G. This mutation changes the codon from CUG to GUG. When the ribosome reaches the mutated codon, it inserts a valine into the growing polypeptide chain. The resulting mutant polypeptide has a valine instead of a leucine at this position and may or may not be functional. Now let's create a mutation by deleting a single nucleotide from the sequence. Notice that the reading frame downstream of the mutation has been altered. In this example, the first altered codon is now UGA, a stop codon, so translation results in a shortened polypeptide, which is likely to be non-functional. What happens if we delete 3 nucleotides? In this example, the deletion of 3 nucleotides preserves the original reading frame, but results in the deletion of 1 amino acid. The resulting mutant polypeptide may or may not be functional. As our final example, let's look at an insertion mutation, in this case an insertion of 2 guanine nucleotides. The insertion results in a new codon, GGC, which codes for a glycine. In addition, a 2-nucleotide insertion disrupts the reading frame, and in this case, the next codon is now UGA, a stop codon. The mutant polypeptide is significantly different from the wild-type version and is likely to be non-functional. Note that an insertion or deletion of one or two nucleotides results in a shift in the reading frame of all downstream codons, whereas an insertion or deletion of 3 nucleotides does not cause a frame shift.